Hello, this is Dr. Sujata Magdum, Assistant Professor in Zoology, Department of Zoology, KTHM College, Nashik. I'll be dealing with Immunology for MSc II, Semester 3, and we are started with the first chapter, that is Introduction to Immune System, and in that I shall be discussing the cells of immune system today. Now, this lecture will take you to certain previous facts what we have discussed earlier regarding immune system. You must be acquainted with the fact that like any other system which is present in our body, immune system plays an important role in the development of immunity of individual. Immune system is comprised of large number of organs and the tissue or the cells which are directly into development of immunity. Further, you must be familiar with the fact that this immune system is made up of two important or distinct organs which are called as the primary and the secondary lymphoid organ. Now, these primary and secondary lymphoid organs basically are concerned with development and activation of immunocompetent cell. Once the immunocompetent cells are developed and they are activated, they are ready to impart immunity. When we are talking about immunity, we need to know about the type of immunity they are involved into. So, we have already studied in previous lecture the two different types of immunity which are being generated into the body by presence of immunocompetent cells, and it can be classified as the innate immunity and the adaptive or acquired immunity. The classification is basically based on the development of immunocompetent cell. The innate immunity forms the first line of defense mechanism, as whatever organs are involved into this type of immunity, they are seen present into the host body since birth, and therefore it is also called as inborn immunity. Whereas there should be additional immunity sometimes required, and this is achieved by the artificial means or from the cells which are not present into the body. Say for example, in presence of vaccine, whatever bodily cells are present, they get activated, and therefore such immunity which is achieved in presence of some extra cells which are not of the own body cells, that forms a part of adaptive immunity. Therefore, adaptive immunity is called as second line of defense mechanism. Now, this innate immunity as well as adaptive immunity, it involves certain kind of cells which can be broadly classified as lymphocytes, that is T and B cell, and also the natural killer cell which is a type of lymphocyte, dendritic cells, Langerhans cells, macrophages, mast cells, monocytes and granulocytes. So all these collectively they form innate immunity, the cells of innate immunity. So why are we calling them cells of innate immunity? Because whatever cells are seen mentioned here, these cells they are seen present since the birth, and therefore they form the part of innate immunity or cells of innate immunity. Now, of these, the T cells and B cells, they are also seen as a part of adaptive immunity because their activity is enhanced. It is increased by addition of certain kind of stimulation from the external means in the form of vaccines, and therefore they form the cells of adaptive immunity. But this is achieved only when there is activation done from the external sources. Therefore, we can say they are a part of adaptive immunity. They are the cells of adaptive immunity. I hope it is clear to you, right? Now, these distinction between the cells of innate immunity and adaptive immunity, we shall be learning shortly in detail. But before that, we need to know the course of development of these cells. These cells, they are seen developed in bone marrow, and the process by which the cells are developed in bone marrow is called as hematopoiesis. The hematopoiesis process involves hematopoietic stem cells, right, which is abbreviated as HSC. Now this HSC is called as multipotent cell. Now before we discuss what is multipotent cell, we'll see the development of this multipotent cell and then we'll continue with the discussion. Multipotent cell, they are developed from pluripotent cell, whereas pluripotent cells, they are seen developed from the totipotent cell, right? So let us see what do you mean by totipotent cell. Toti refers to complete or total. Now, it means that this totipotent cell has the ability to give rise to any type of bodily cell present into the organism. Now, the best example to describe the totipotent cell is zygote. You all know that zygote is formed by fusion of sperm and ova, and this zygote, it has the ability to develop into a multicellular organism which contain different types of cells. Now, these different types of cells which are present in an individual, they are seen developed from the zygote, and therefore it is called as a totipotent cell. Or in other words, I can say that this zygote, it has the ability to give rise to various different bodily cells as well as the placental cell in the early embryonic stage of development, right? Thus, it is called as a totipotent cell. Pluri refers to presence of stem cells which can develop into some type of cells. Means it can give rise to large number of cells except to the placental cells. Therefore, it is called as pluripotent cells. Pluri refers to some. So, distinction between toti and pluri is toti can give rise to pluripotent as well as placental cell, whereas pluripotent cells, they can only give rise to the bodily cells, whereas it cannot form any placental cell. Therefore, it is called as a pluripotent cell. Now, these pluripotent cells, they are also called as embryonic stem cells, and these embryonic stem cells later on get differentiated into the multipotent cell. Now, once again, if I split this multipotent word into two, we have multi and potent as two different words. Multi gives the meaning as many. That means this multipotent cell has the ability to get differentiated into many types of cell. Like it can give rise to lymphoid cell as well as the myeloid progenitor cell if we are talking with reference to hematopoiesis, which is seen present in the stromal tissue of bone marrow, which contains hematopoietic stem cells. Therefore, hematopoietic stem cells, we can say they are multipotent cell. Now, once these multipotent cell undergo the process of differentiation as the lymphoid progenitor cell or the myeloid progenitor cell, then these cells or these progenitor cells, they are distinct in giving rise to only one type of lineage or one line of cell development, that is called as lineage. Therefore, we can say that HSC, which is a multipotent cell, will get now transformed into unipotent cell, and this unipotent cell will form two lineages, that is lymphoid progenitor lineage as well as myeloid progenitor lineage, indicating that the lymphoid progenitor cells or lineage can give rise to only lymphoid cells. Now, what are different types of lymphoid cells? In the earlier slide, we have seen that lymphoid cells include T cells, B cells as well as the NK cells or natural killer cell. Whereas myeloid progenitor cell, it is involved into formation of the blood cells. That means it can give rise to both the RBC, WBC, or even it is involved into formation of the platelets. Therefore, this lymphoid progenitor cell, as it gives rise to only lymphocyte, we can say it is unipotent cell. Whereas myeloid progenitor cell, as it is involved into formation of blood cell, it can be called as a unipotent cell. Right? Now the next, we'll see how the course of development occurs. Now, as in the earlier videos, you might have come across the concept of the bone marrow. You know that bone marrow is a part seen only into the long bones. The stromal tissue of this bone marrow, it contains large amount of the stromal mass consisting of fibroblast, mast cell, macrophages, or you can say different types of cytokines, or you can call it as the proteins which are essential for development of immunocompetent cell. Now these different additional components which are present into the stromal tissue, it provides a microenvironment wherein the hematopoietic stem cell can get differentiated into the myeloid as well as the lymphoid type of tissue. Now this myeloid and lymphoid type of tissue which is seen developed into stromal tissue, it is under the influence of erythropoietin or the cytokines, which are commonly called as growth factors or growth regulators. Now these growth factors or growth regulators, they enable the HSC, that is hematopoietic stem cell, to undergo differentiation, and this differentiation leads to formation of the myeloid as well as lymphoid progenitor cell. There is different steady state regulation observed into the process of hematopoiesis, and it totally depends on the level and the type of cytokines which are present into the stromal tissue. Further, the regulation of hematopoiesis is also dependent on the production of cytokines and the hematopoietic activity between the T and the macrophage cells. Regulation of expression of receptors for hematopoietically active cytokines in the stem cell and progenitor cell also aid in regulating the hematopoiesis in bone marrow. Further, removal of some cells by controlled induction of dead cells from the stromal tissue is also regulated for hematopoiesis. Now these immunocompetent cells what we are discussing, they need to understand or distinguish between the bodily cells and the infected cells or foreign cells. So what makes the immunocompetent cell to distinguish this? We will try to understand. Now here, try to observe what I am focusing. This is NK, refers to natural killer cell, which is one of the type of immunocompetent cell. There is another healthy cell of the body, and this healthy cell, you can see it shows presence of large number of receptors onto its surface. This is the common property, you can say, or the ideal property of any bodily cell present into our body. There are different specific receptors which are seen present onto the body cell. Right? Suppose there is a pathogen which has made its entry into the vicinity of healthy cell. Obviously, this pathogen by its tactics makes its entry within the cell, that is healthy cell. Once the pathogen enters into the healthy cell, what distinction you can make between the healthy cell and the infected cell? If you observe, the infected cell, it does not show presence of any receptor molecules on its surface, right? As a result, the natural killer cell, when it comes across such infected cell without any receptors on its body, will identify the infected cell as a harmful cell, and it will trigger a response which will bring about lytic action, and so the infected cell will get fragmented or it will be broken down. Once this is broken down, what we can say, we have achieved immunity, right? Isn't it? Can you see this infected cell which is now totally losing its identity, and that is because it got fragmented, and this fragmentation or lytic action is because the natural killer cell has identified this particular cell as an infected cell. And why this natural killer cell can identify this as infected cell? Because the cell surface, it totally lacks the receptors which otherwise are seen present onto the surface of the cell. Correct? Sometimes what happens, the infected cell will release its secretion outside the surface, in the sense like, suppose the pathogen is a virus. So you are familiar with the fact that viruses, they will use the machinery of the host cell nucleus, and they will start synthesizing the proteins, and those proteins, they will be expressed onto the surface of the host cell. Now this diagram, what it is depicting, you can see the nuclear component where the orange part which I am focusing is indicating the viral genome, whereas the thinner maroonish thread-like structure is a part of healthy cell. Obviously, the viral genome will utilize the machinery of the host cell, and by the action of transcription and translation, the viral genome will synthesize large number of proteins which will be expressed onto the surface of the host cell. Correct? Now, if you see this protein which is present on the host cell, it is totally different from the bodily cell's receptors. As a result, once again, the natural killer cell will identify this cell as an infected cell, and it will bring about the lytic action, or you can say it will cause the fragmentation. Hence, we can say that this way the immunocompetent cell provides immunity or helps in development of immunity. Now, this immunocompetent cell is totally opposite to the terminologies like immunodeficiency, immunoincompetent cells, or immunocompromised cells, which are commonly used in the textbook of immunology. So let us see what is immunodeficiency cells. Now, immunodeficiency refers to the cells which are not showing or which are not capable of showing immunity against infection. The best example is the newborn. You all know that in newborn, immunity level is low and they are prone to get infected more easily as compared to adults. Now, this is only because the cells are immature, or probably the number of immunocompetent cell into the body, they are less in number. Whereas in their circulation, in the body of these newborn, there are some ready-made maternal antibodies which are circulated. They provide immunity till the newborn acquires developed immunocompetent cell. Now this is the what you can say plus point of the nature which enables the newborn to sustain life healthily. The second terminology which is relatively used in immunology is immunoincompetent cell. Now here, incompetency is with reference to failing of immune system to show the immune action. That means the immunocompetent cells, they are probably not able to overcome the action of the pathogen, as a result of which they cannot identify the infected cell, neither they can identify the cells with certain kind of differences onto their body, onto their surface, which is not seen into the bodily cell. Therefore, in such cases, the immune system fails to recognize the pathogenic cells or the infected cells, and therefore the pathogen they flourish. The best example is AIDS, which we say acquired immunodeficient syndrome. So it is because the cells are not able to recognize the immuno suppressing units like the pathogens. Therefore, in AIDS patient, many different types of infections are seen commonly occurring, and therefore it is called as acquired immunodeficient syndrome. In the same way, the third terminology is immunocompromised cells. Now, compromised refers to acceptance of whatever is forcibly added into the body. Example is like you might have come across many times the word transplantation. So what actually occurs in transplantation is there is the organ or the tissue which is not belonging to the host body is forcibly introduced into the deceased body so that there will be sustaining life into the host. Now, for this, the cells of the host body, they compromise and they accept the organ which is foreign to them, and the organ sustains its viability into the transplanted individual. Therefore, such cells which help in accepting the donated organ from other body, they are called as immunocompromised cells. Now, this immunocompetency is achieved by activation. Now, how this activation occurs? For this, you need to understand the distinction between the naive cells and the activated cell. So, what is naive cell? Naive cells are those cells which have never come across any antigenic stimulation. So, such cells which are not seen, acquainted with any foreign particle, they are defined as naive cell. These are very small, motile and non-phagocytic in nature, and their size is very small, which measures nearly 6 micron in diameter. Such naive cells, they are always seen present into the bone marrow. Once these naive cells come into circulation, and if by chance they get acquainted with some foreign particles or antigen, then the naive cells get activated into the mature cell. And during maturation, they attain the size which is measurable around 15 micron in diameter. Now let us see how this happens. All the naive cells which are present into bone marrow, they form nearly 20 to 40% of body's white blood cell and 99% of the lymphatic secretions. There are three different types of these lymphocytes which are present into the body, and they can be classified as T, B and natural killer cell. T and B cell, the name is thymus cells or T cells, bone marrow cells or B cells, and natural killer lymphocyte. T and B cells, they get activated. As I said, they should be educated or they get antigenic stimulation, and so this antigenic stimulation makes the naive cells get activated. If the activation occurs into the thymus, they are called as T cell. If it occurs into the bone marrow, it is called as B cells. Right? So, when we are talking about this process of activation, please concentrate onto the diagram. Here, the cursor is showing the nave cell, which is small, and probably the diameter is 6 micron. Now, such nave cells, they are always seen in G0 phase. That means these cells, they are into the resting phase. So such resting phased cells or lymphocytes that come into circulation through the blood or into the lymph, and during the course of circulation they get settled into secondary lymphoid tissues, right? So when they either into the circulation or when they are seen present into secondary lymphoid organs, that time if there is an antigenic stimulation, it induces the inactive resting cell to enter into the next stage of its cell cycle. As I said, this small nave cell is in G0 resting phase. Now, because of antigenic stimulation, it will enter into the G1 phase. Later, this will get converted into, or it will enter into G2 phase, and then leads into the mature phase or comes into the M phase, that is the mitotic phase, wherein it undergoes the process called as division. This division will lead to formation of different types of cells like the effector cells or plasma cells. Effector cells or plasma cells are one and the same type of cell, and the other type of cell which is formed is the B memory cell. Now here the example which is shown into the diagram, it is with reference to B cell. That is why the B cell on antigenic stimulation will get converted either into the plasma cell, or it has the chances to get converted as the memory cell. So plasma cells, they are involved in synthesis of the antibodies, whereas memory cell, which will once again be seen into the resting condition, will move into circulation and will be utilized if the same antigen once again makes its entry into the body. That means the second stimulation from the same type of antigen will not help in taking much more time for converting itself to memory cell or to effector cell. Because there is presence of memory cell, the second antigenic load will immediately stimulate the memory cell, and these memory cells will get transformed into plasma cell, so that the plasma cells will start synthesizing the antibodies, and therefore there will be development of immunity against any pathogen which has made its entry. Right? Now the lower diagram shows the different developmental stages of B cell. The first phase is the nave cell. That means here the cell size is small. It shows a very large nucleus. Then the cytoplasm is forming a rim-like structure. Therefore, we can say endoplasmic reticulum, Golgi complex, mitochondria, they are located into the peripheral region. And the volume of the cytoplasm is very less. If you compare the volume of cytoplasm to the next stages, that is the active stage or the plasma cell, we can make out that the cytoplasm volume is much more higher into the blast cells or into the plasma cells. Now what are blast cells? The intermediate stage. Once this nave cell gets an antigenic stimulation from G0, it gets converted to G1 phase. So conversion of G0 to G1 phase helps in identifying the nave cell getting transformed into the blast cell. Now this blast cell is also called as lymphoblast cell. This lymphoblast cell which has entered into G1 will now get transformed into G2 and will enter into the phase of maturation, leading to the cell division and helps in converting the lymphoblast into the memory cell or into the plasma cell. So if it gets converted into the plasma cell, then this plasma cell will start synthesizing large amount of antibodies against the foreign particle which has stimulated the naive type of naive type of bodily cell. Thank you.